everyone. Welcome to our today's session. Here myself, Dr. B. N. Thorat, Assistant Professor, Department of Animal Husbandry and Dairy Science. So in that today's theoretical session, we have to see the Mendelian principles and Hardy-Weinberg law. This is our fifth number topic. So in that topic, always asking 4-mark questions or 1 or 2 objective in your final theory examination. Okay, so what is the Mendelian principle or how much Mendelian principles we are taught in our this topic and which are the Hardy-Weinberg law. So in that Mendelian principles, Mendelian has worked on a different 7 traits. So result of crosses from parent to F1 generation, their pattern of transmission of hereditary particles. So in that there are different laws. So first law is a law of segregation. Segregation means separation. So what stated this law? Law of segregation is stated when a pair of contrasting character which can occur together in a hybrid, that means F1 generation, they remain together without mixing with each other and segregate during the formation of gametes. So this is the example of law of segregation. So in that law of segregation, the mating of pure breeding, it should be polled bull. It can be denoted by P alphabets to the horned cow, that is small p alphabets. It should be produce F1 generation and mating between F1 to produce the F2 generation. So here there are some parent, that means phenotypic we have already seen, that is polled bull and horned cow. So genotype of that is capital P and horned cow, that is small p. Their gamete, it should be divided at the time of segregation, so it should be capital P and small p. Whenever they cross and make a F1 generation, so with what is the F1 generation? Capital P, small p, it should be polled. Okay, then again this F1 generation is crossed with a F1, that should be capital P, small p and capital P, small p. So which is the phenotypic ratio of that law of segregation? That is 3:1, and genotypic ratio, it should be 1:2:1. So in case of law of independent assortment, that is the second law. The law of independent assortment, it should be stated the segregation of one pair of allele, it should be independent of the segregation in any other pair of allele. Okay, it should relate the simultaneous segregation of 2 or more genes that control 2 or more traits. That means dihybrid and dihybrid inheritance. So what is meant by dihybrid? Dihybrid is a cross which involve 2 pair of contrasting traits, and dihybrid inheritance means inheritance associated with the 2 traits. So example of law of independent assortment, it should given there are 2 types of traits it should be used. One is polled and another is a horn. Polled, it should be denoted capital P, and horn, it should be denoted small p. And another traits is a black color and red color. Okay, so black polled bull is crossed with a red horned cow, they should produce F1 generation, and again this F1 is crossed with the F1, they should produce F2 generation. So parent, that is phenotypically ratio or phenotypically parents is black polled crossed with a red horn. Okay, so this is given in that chart. So their phenotypic ratio is 9:3:3:1, and genotypic ratio is 1:2:2:4:1:2:1:2:1:2:1. That is black polled and red horn got broken down during the gamete formation and got combination again at a fertilization. Thereafter F2 generation or F2 exhibited some new combinations which were not present in a parental or F1 generation, that is black horn and red polled. Then again backcross. In the backcross means mating of F1 to either one of the 2 parental type. So in case of backcross, the parent is a phenotypic, that means polled and horned. Okay, so in that genotype we have to be used for polled capital P, and in case of horn small p, small p. So their gamete capital P and small p, they have to be produced F1 generation, that is capital P, small p is the polled. Then again backcross, one of their parent out of two. So this F1 cross with a polled, so they have to be capital P, small p, then capital P, capital P. So their gamete capital P, small p and capital P. Okay, so they have to be produced capital P, capital P, capital P, small p, that is all polled. Okay, so these type of backcross, that means they have to be mating F1 either one of their parent. Then in case of test cross, that means mating of individual of incompletely known genotype to the individual of homozygous recessive genotype. Then another, Mendel's law of segregation. So in that Mendel's law of segregation, in modern term, that should be stated that a diploid individual which carries two individual copies of each autosomal gene, which are that one copy of each member of a pair of homologous chromosome. Each gamete, it should be produced by a diploid individual. The recessive, only one copy. You should remember, recessive or individual receive only one copy of each gene, which is chosen at a random from the two copies and found in that individual. Under the Mendel law of segregation, each of two copies in an individual has an equal chance to begin include in a gamete, such that it should be expect 50% of an individual. So this is a diagrammatic representation of your law of segregation, that is in circle there are two types of a heterozygous parent. The one is capital A, another is a small a. Whenever they are segregate of allele into the gametes, 50% possibility of dominant and 50% probability recessive. So these are segregate from each other and produce capital A, small a, that is their heterozygous gamete. Then which are the Hardy-Weinberg law, or what it should be stated? For Hardy-Weinberg law, it is also known as a Hardy-Weinberg equilibrium or Hardy-Weinberg model or Hardy-Weinberg theorem, as well as Hardy-Weinberg law. These all are the same or synonymous. Whenever likewise questions which will be asking in your examination, it should not be confused, but answer is the same for Hardy-Weinberg principle, Hardy-Weinberg equilibrium, Hardy-Weinberg model, Hardy-Weinberg law or theorem. All of you are remember that point. The principle stated, the allele and genotype frequencies in the population will remain constant from generation to generation in the absence of other evolutionary influence. So this Hardy-Weinberg law, it should be in a large random mating population, the gene and genotypic frequencies remain constant from one generation to the next in the absence of migration, mutation and selection. All of you are remember this point. The gene and genotypic frequencies remain constant from generation to generation. A population which fulfill the condition of this law is said to be Hardy-Weinberg equilibrium or Hardy-Weinberg law. In or the population in Hardy-Weinberg equilibrium, it should be exhibit a distance relationship between gene and genotype frequencies. It should be given below. If we have considered small p and small q are the respective frequencies of the allele, and capital A and small a among the parent, then the frequencies of a different genotype in Hardy-Weinberg population, it should be occurs different. That is frequency of homozygote dominant, that means capital A capital, it should be P. Then frequency of homozygous recessive, that means small a, it should be denoted Q. And frequency of heterozygous, that means F1 capital A and small a, it should be 2pq. And which are the different assumptions or factors they are affecting this Hardy-Weinberg law or Hardy-Weinberg equilibrium? Okay, so first one, large population. In that large population, it should be indefinitely large, which is in a practical term means that this population is large enough such that the chance does not play any type of role in changing the allelic frequency. Then second one, for practical purpose, population is considered large if the number of individual is in a 100 or 1000. Then another, random mating. In random mating, mating within the population for a genotype under the consideration occurs at an random. Then third one, no selection. Whenever no selection, equal visibility, equal viability and reproduction of a different genotype in the population. Then no migration, there is a no movement of an individual from or into the population, that is closed population, and no mutation, so there is no mutation from the allelic state to the another. Okay, and another most important part, that is application of Hardy-Weinberg law. On this topic, likewise question is asking, state or write down Hardy-Weinberg law and write down its application in genetics. So application is most important part. First application, the study of genetic stability of population. Then second one, prediction of allelic and genotypic frequency under the complete dominance. Third application is prediction of future, that means progeny, genotype frequencies. And the last application of Hardy-Weinberg law, that is prediction of genotype for a genetic counseling. So thank you all of you for your patience. Thank you all of you.